this lecture about hemoglobin and myoglobin, which is lecture 4 in session 2 in molecular gene and disease module, presented by Dr. Zainab Al-Manasir. The learning objective of this lecture is a continue to the learning objective of lecture 3. So, in this lecture, we try to explain the physiological rules of myoglobin and hemoglobin, contrast the oxygen binding properties of myoglobin and hemoglobin, and explain why hemoglobin is most suited to its role as an oxygen transporter. Also, describe the major structural differences between oxygenated and deoxygenated hemoglobin and the molecular basis of cooperativity. Learning objective number 7 is to describe the effects of carbon dioxide, hydrogen, 2,3-bisphosphoglycerate, and carbon monoxide on the binding of oxygen by hemoglobin, and the physiological significance of these effects. The last learning objective is about the mutations in globin genes which can give rise to diseases such as sickle cell anemia or thalassemia. Hemoglobin is an iron-containing protein that transports oxygen in human blood from the lungs to tissue of the body. So, hemoglobin means heme plus globin. Each hemoglobin molecule is made up of 4 units of heme, so the green color units is a heme unit or heme group, iron group, surrounded by a globin protein groups. So what is heme? Heme is an iron porphyrin compound, and porphyrin is a tetrapyrrole structure. These are pyrrole structure. The ferrous iron occupies the center of the porphyrin ring and establishes 4 nitrogen bonds of all the pyrrole rings. Therefore, iron attached to 6 coordinated bonds, 4 coordinated bonds planar with the nitrogens, 1 coordinated bond linked to oxygen, and 1 coordinated bond linked to histidine amino acid of alpha or beta globin chain. The central iron provides reversible binding to oxygen and carbon dioxide molecules. So this is the structure of a heme, which consists from 4 pyrrole rings. In the center there is an iron, and this iron makes a linkage with the 4 linkage with the nitrogen, and 1 linkage with the globin, with the amino acid histidine that present in the globin, alpha and beta, and also 1 link with the oxygen, and this linkage is reversible, which means it can be bind with the oxygen and bind with the carbon dioxide. So heme groups can be present in other compounds rather than hemoglobins, like myoglobin, cytochromes, and peroxidase. Globin, hemoglobin comprises 2 pairs of globin chains that are twisted together, and in normal adult hemoglobin there are 141 amino acids in each alpha chain and 146 amino acids in each beta chain. Therefore, the complete structure of the hemoglobin is 4 heme units surrounded by 4 globin units, and each heme can carry 1 molecule of oxygen. What are the main types of hemoglobin? The first one is hemoglobin A1, and this is alpha 2 beta 2, represent about 97% of total hemoglobin, and consisting of 2 pairs of globin, which are alpha pair and about 141 amino acid residue, and beta pair 146 amino acid residue. The other types of hemoglobin is hemoglobin A2, and this is represent about 2 to 3% of the total adult hemoglobin. It consisting of two pairs of globin alpha 2 and delta 2. Third type of hemoglobin is hemoglobin F, which is a fetal hemoglobin. This hemoglobin consisting of alpha 2 and gamma 2 is present primarily in the embryonic life and usually disappear from the circulation by the age of 6 months. Hemoglobin F represents less than 1% of the hemoglobin in adults, and it has a greater affinity for oxygen than adult hemoglobin. The last type, or the fourth type of hemoglobin, is GAWAR1 and 2. These are synthesized by the embryonic yolk sac during the first month after conception. For hemoglobin E GAWAR1, consisting of zeta 2 and epsilon 2, while hemoglobin E GAWAR2 consisting of alpha 2 and epsilon 2. This figure showing that the three types of the hemoglobin, the embryonic hemoglobin, fetal hemoglobin, and the adult hemoglobin. This table shows the difference of the hemoglobin in the composition and in the percentage. So for the hemoglobin A1, it composed of alpha 2 and beta 2 and represent about 97% of the total hemoglobin. Hemoglobin A2 consisting of alpha 2, delta 2 and represent about 2%. Hemoglobin F consisting of alpha 2, gamma 2 and represent about 1% of the adult hemoglobin, while at birth is about 80%. And as I mentioned before, that hemoglobin F disappear from the circulation by the age of 6 months. Regarding globin synthesis, globin is synthesized by the ribosomes in the cytosol. And there are two genes located on chromosome 16 responsible for encoding of the alpha 2 chain, while the other 4 genes located on chromosome 11, they are responsible for the encoding. One gene for the beta chain and one gene for the delta chain and 2 genes for the gamma chain. So in the embryonic hemoglobin, which is GAWAR1, this consisting of zeta 2 that present on chromosome 16 and epsilon 2 present on the chromosome 11 that encoded from the gene present or located on chromosome 11. While GAWAR2, alpha 2, this is transcribed from the gene alpha gene that located on chromosome 16 and epsilon 2 transcribed from a gene located on chromosome 11. Regarding fetal hemoglobin, hemoglobin F, consisting of alpha 2 and gamma 2. Adult hemoglobin, either hemoglobin A1, this is alpha 2 beta 2, or hemoglobin A2, which is alpha 2 delta 2. Regarding heme synthesis, heme synthesized in the bone marrow and in the liver. So their site of the synthesis is in the mitochondria and partly in the cytoplasm. The substrate for heme synthesis is succinyl-CoA plus glycine, and the pathway or the process for heme synthesis consisting of 8 steps. Myoglobin is a polypeptide, is a single polypeptide, structurally similar to the hemoglobin, but the hemoglobin is tetramer, myoglobin is monomer. So, because it is a single polypeptide, it can bind only to one molecule of oxygen with high affinity. And the function of the myoglobin is for storage of oxygen inside the muscle and to be used later on when the partial pressure of the oxygen be very low. This table reveals the difference between hemoglobin and myoglobin. So the number of chains in the hemoglobin, 4 polypeptide chains. In myoglobin is single polypeptide chains. Therefore, hemoglobin is a tetramer, while myoglobin is a monomer. Hemoglobin can bind to the carbon dioxide, carbon monoxide, nitroxide, oxygen, and hydrogen, while myoglobin binds to oxygen tightly and firmly. Hemoglobin presents systemically in all over the body, while myoglobin in the muscle cells. The type of the curve of the oxygen dissociation curve for hemoglobin is a sigmoidal in shape, while in the myoglobin is hyperbolic curve. The abbreviation of hemoglobin HP and the myoglobin MB. The role of hemoglobin is to carry the oxygen, while the myoglobin is to store the oxygen. Concentration of hemoglobin in the RBC is high, while the myoglobin is low. Hemoglobin molecule can exist in two forms or in two states. The first one is the T-tense state or TOT. This is a deoxyhemoglobin, which means that lacks of the oxygen, and here the globin units are held tightly together by electrostatic bonds. The other form of the hemoglobin is the R state or relaxed, and this is in oxyhemoglobin, which means that fully oxygenated. The bonds here are broken when they are bound to oxygen, and it has a high affinity for oxygen than in T form. Cooperative binding of oxygen to hemoglobin. This features is to change the conformation of the hemoglobin or change the state of the hemoglobin from the T state to R state. So, when oxygen molecule binding to the hemoglobin, to the first heme, this will lead to altering in the conformation or alters the conformation of the hemoglobin that allow the other units to bind with oxygen, which means that change the state of the hemoglobin from the T state to R state in order to bind more oxygen. And conversely, when each molecule of oxygen released from the hemoglobin, this will change in the shape or in the conformation of the hemoglobin and make it easier to other oxygen to release from the hemoglobin, which means that conversely, each oxygen molecule is released. The change in hemoglobin makes it easier for other molecules to be unloaded. The oxygen dissociation curve, it's a curve that plots the proportion of hemoglobin in its saturated form on the vertical axis against the partial pressure of the oxygen on the horizontal axis. And as you see here that for the hemoglobin, the curve of the oxygen dissociation is sigmoidal in shape, while the curve of the myoglobin is hyperbolic in shape. There are certain factors affecting the oxygen dissociation curve of the hemoglobin. First of all, if shift the oxygen dissociation curve to the right, this means that the hemoglobin has low affinity to oxygen, and this effect we call it Bohr effect or known as Bohr effect. This effect is a response to high temperature, high PCO2, high partial pressure of carbon dioxide, in case of decrease in the pH, and increase in 2,3-diphosphoglycerate, and the other name is 2,3-bisphosphoglycerate. While if the curve of the oxygen dissociation curve shifting to the left, this means that the hemoglobin has a high affinity to oxygen, and this effect we call it or known as Holden effect. This effect is a response to the low temperature, low partial pressure of carbon dioxide, high pH, and low level of the 2,3-diphosphoglycerate, and in case of carbon monoxide poisoning. So what's Bohr effect? Bohr effect is a decrease in the oxygen affinity of a hemoglobin, and this occurred in response to increase in the temperature, increase in the partial pressure of the carbon dioxide, and increase in the 2,3-diphosphoglycerate. Also in case of low pH. 2,3-diphosphoglycerate, this is a special intermediate of glycolysis in erythrocyte, and this plays an important role in enhancing or in liberating of oxygen from the hemoglobin. So this is a dissociation curve, oxygen dissociation curve. This is the vertical axis represent the oxygen saturation of the hemoglobin, while the horizontal axis represents the partial pressure of the oxygen. So you see here that at 100 millimeter mercury, and that is in the lung, the oxygen saturation of the hemoglobin about 100 or less than 100, 98 to 100, and this is for the normal curve, for the normal person in resting state. While in case of exercise or in the contraction of the muscles, we see that increase in the temperature and increase in the partial pressure of the carbon dioxide, and this will lead to the increase in the hydrogen ions, and the hydrogen ions lead to the decrease in the pH, and at the same time increase in the 2,3-diphosphoglycerate. So these factors will affecting the curve and causing shifting of the curve to the right side. So this means that here the hemoglobin become less affinity to the oxygen, and it's ready to liberate of the oxygen to the tissue. In Holden effect, it's the opposite to the Bohr effect. Is an increase in the oxygen affinity of the hemoglobin, and this promotes the release of the carbon dioxide in response to decreased temperature, and decrease with carbon dioxide, increase with pH, decrease with 2,3-diphosphoglycerate, and in case of carbon monoxide poisoning. So again, this is an oxygen dissociation curve. The vertical axis represents the oxygen saturation of the hemoglobin, the percentage of the oxygen saturation of hemoglobin, and the horizontal axis represents the partial pressure of the oxygen. So what happened here? In case of low temperature or low carbon dioxide, and in case of high pH and low 2,3-diphosphoglycerate, and also in case of high carbon monoxide poisoning, the curve will be shifted to the left, and this means that the hemoglobin has a high affinity for oxygen, which means that the hemoglobin is not ready to release the oxygen. It will keep the oxygen، يعني يحتفظ بكمية الأكسجين الموجودة ما يطرحها. So this is called the Holden effect. So there are two questions here. Why 2,3-bisphosphoglycerate decreases the oxygen affinity of hemoglobin? And why patient with carbon monoxide poisoning has a cherry red or pink cheek appearance? The answer of the first question is because 2,3-biphosphoglycerate, bisphosphoglycerate, decrease the oxygen affinity of the hemoglobin because this compound binds with the deoxyhemoglobin but not with the oxyhemoglobin. 2,3-bisphosphoglycerate binding with the deoxyhemoglobin and not to the oxyhemoglobin. This binding will stabilize the deoxyhemoglobin in a T state, يعني keep the hemoglobin in a tense state, in a T state, so not allow the oxygen to, so for that reason it will be decrease the oxygen affinity. So this means that it prevents the heme site to bind with the oxygen. While in carbon monoxide poisoning, the answer for the carbon monoxide poisoning, because carbon monoxide, when it's bind to one or more of the heme molecules or heme site, this will lead to the shifting of the hemoglobin from the tense state into a relaxed state. This shifting from the tense to the relaxed, causing that the remaining site of the heme to bind with the oxygen with high affinity. The other question is why hemoglobin F has high oxygen affinity than hemoglobin F has high oxygen affinity than hemoglobin A. This curve showing that the adult hemoglobin and the fetal hemoglobin. What I mean here that the oxygen dissociation curve of the fetal hemoglobin and the oxygen dissociation curve of the adult hemoglobin. The answer for this equation is that because hemoglobin F having a gamma subunit, and this gamma subunit allows to bind more strongly with the oxygen. While the second answer is because the presence of 2,3-bisphosphoglycerate. This compound interact much more with the hemoglobin A than hemoglobin F. Why? Because the adult beta subunit has more positive charges than the fetal gamma subunit. And these positive charges attract the negative charges from the 2,3-bisphosphoglycerate. Therefore, the preference of 2,3-bisphosphoglycerate for hemoglobin A makes that the hemoglobin F binds to oxygen with high affinity. Hemoglobin abnormality, which is a variant form of hemoglobin that is often inherited and may cause a blood disorder. These disorders could be in the heme production and that lead to the porphyria, or could be in the globin synthesis, and globin synthesis either affecting the structure, for example sickle cell disease, or affecting the production of the globin like thalassemia. Porphyria is a group of disorders that caused by abnormalities in the chemical steps that lead to the heme production. So as I mentioned before that synthesis of the heme is occurred by 8 steps, part of them in the mitochondria and part of them in the cytoplasm. So any defect in each one of these 8 steps will lead to the disease or lead to the defect in the heme production. So, and this will lead to the porphyria. Porphyria, there are many types, but the most common type of porphyria are acute intermittent porphyria, and the second type is porphyria cutanea type. Regarding the abnormality in the globin synthesis, and as I said before, in the structure which lead to the sickle cell anemia, and this caused by a mutation in the beta globin gene that change the position of the amino acid, the 6th amino acid, from the glutamic acid to valine. Glutamic acid, this is a charged amino acid, to valine, which is a non-polar amino acid. Normally, in a normal hemoglobin, the glutamic acid, or the sequence of the glutamic acid, is the 6th. It's the 6th amino acid. In sickle cell anemia, this 6th amino acid replaced by valine. So sickle cell anemia could be homozygotes, which means that produced only abnormal beta chain, and that lead to the hemoglobin S, or heterozygotes, which produce a mixture of normal and abnormal beta chains, and that makes normal hemoglobin A and hemoglobin S. The pathology of sickle cell anemia is when hemoglobin S is deoxygenated. So the molecules of hemoglobin will polymerize, forming structures or bodies called pseudocrystalline structures, these known as tactoid. Tactoid will distort the red cell membrane and produce a characteristic of sickle-shaped cell. So this is a normal RBC. When this hemoglobin becomes deoxygenated, these RBCs, the hemoglobin in these RBCs polymerize, forming pseudocrystalline bodies called or known as tactoid, and these tactoid lead to the damaging of the membrane of the red blood cells, forming a characteristic of sickle cell or sickle-shaped cells. Characteristic of sickle-shaped cells. While in thalassemia, there is defect in the production or in the rate of a production in one or more of the globin chains, and this leads or result to the imbalance in the globin chain synthesis. So for the example of thalassemia could be alpha thalassemia, and this means that there is a defect in the synthesis of the alpha globin chain, or beta thalassemia, defect in the synthesis of the beta globin chain. In alpha thalassemia could be single gene mutation, that means mutation in a single gene, and this is clinically silent, or 2 gene mutations, and this lead to the microcytic anemia, or could be 3 gene mutations, which lead to the HBH disease. In this case, beta chains will precipitate as beta-4 tetramers, which means that HBH, and this lead to moderately severe conditions of alpha thalassemia. The last one is 4 gene mutation. The other name for it is hydroxyfetalis, and this lead to the forming of the gamma-4 tetramers, which this is an HB parts, and lead to the hydroxyfetalis. In this case, it is incompatible with life, or the fetus here is incompatible with life. So in case of beta thalassemia, mutation in the beta globin gene, this leads to the generation of a stop codon in the messenger RNA. This stop codon will terminate the globin chain synthesis and lead to the variable reduction in beta globin output. So beta thalassemia could be thalassemia major, the other name is Colles anemia, and this is a severe transfusion dependent, which means that the patient needs frequent transfusion of blood. Or it could be thalassemia intermedia, which means that intermediate in severity. Or the third type is thalassemia minor, and this is a symptomless carrier state. Thank you very much for your listening, and If there is any question or confusion, please don't hesitate to ask. Again, thank you very much.